Moving to our topic, what is a cell made up of, right? Cell is mainly made up of certain cell organelles. But where are these organelles present? These are not randomly located anywhere. We have discussed two types of cells. One is prokaryotic, another one is eukaryotic. We know prokaryotic are primitive, means not well defined. And when we talk about eukaryotes have an elaborate internal organization. Means it has a plasma membrane. It has a plasma membrane. It has a membrane bound nucleus. Alright. That nucleus material or this nucleus one is present in the cytoplasm. Together these two are known as protoplasm, the living content of the cell. Alright. When we talk about that how do a cell looks like under a microscope, we see these three things: this plasma membrane, this nucleus, and this cytoplasm is observed. And all the activities inside a cell and the interactions of the cell in its environment is possible due to these features only. Alright, taking up the first topic, plasma membrane. What is a plasma membrane? In eukaryotes, this is the outermost covering which is present in the form of numerous finger-like projections also, which we call villi. Alright, so this is the outermost covering. Why? Because it protects the cell from the external environment. Number two, it separates the contents of the environment of the cell from the environment. Number three, it allows the entry and exit of certain materials in and out of the cell. It is not allowing everything to move out or everything to move in. It also prevents the movement of certain those bacteria and other pathogens penetrating into the membrane. So, then the question comes, why do we call it selectively permeable membrane? Only because it is not permitting the entry and exit of each and every organism which tries or simply implies to gain entry into the cell. If it is so, if it is not allowing each and every cell to take an entry, then how the movement of substances take place into the cell and how do they move out? Like there are special substances, CO2, oxygen, which has to cross across the cell membrane. How do they move? Because CO2 is the excretory gas. Oxygen, which is required by the body, they move because of diffusion, a special process which is a spontaneous movement of substances from a region of high concentration to a region of low concentration. We are quite familiar with this definition. Then, if something similar happens, like this kind of environment is there in a cell, how will it work? I am taking an example of CO2 and oxygen exchange in our body cells. When this kind of example is to be taken into account, we say this cellular waste carbon dioxide, it accumulates in high concentration inside the cell. In the cell's external environment, this concentration of CO2 is very low, but it is high here inside the cell. As soon as there is a difference of concentration inside and outside the cell, the CO2 moves out of the cell. Okay, from a region of high concentration to a region of low concentration by the process of diffusion. Alright. Similarly, oxygen enters the cell by the process of diffusion because oxygen is mainly present in high amount in the external environment. So what will this oxygen do? This oxygen will simultaneously move from a region of high concentration to a region of low concentration. Okay, so this diffusion plays an important role in gaseous exchange between the cells as well as the cell and its external environment. Same way we say water also obeys the law of diffusion. When water moves from a region of high concentration to a region of low concentration through a semi-permeable membrane, we call this process osmosis. Are we familiar with this process? Yes, because the diffusion of water molecules across the plasma membrane is known as osmosis because it is affected by the amount of substance dissolved in water. Osmosis is always dependent upon the amount of, I'm writing it here, amount of substance dissolved in water. Okay, so what is osmosis? It is the passage of water from a region of high concentration, high water concentration to a region of low water concentration through a semi-permeable membrane. Clear? Now we are going to discuss what will happen if we put an animal cell in a semi-permeable membrane. Okay, that is in a solution of sugar or salt. I am taking 3 diagrams simultaneously in detail and taking 3 different topics. My question is, what will happen if we put an animal cell or a plant cell into a solution of sugar or salt? Clear? If the medium, now I have taken this beaker. In this beaker, I have taken a cell. Clear? This medium surrounding the cell has a high water concentration. High water concentration. Alright? Then the cell. It means that the outside solution, which is having a high water concentration, it's more dilute. Clear? It's more dilute. Now, the question arises, if it is more dilute, what will happen to the water which is present outside? Here, high amount of water. Okay, meaning the dilute condition compared to the environment present inside the cell. Obviously, what happens every time whenever this happens, water molecules tend to move in both the directions because they are free to pass across the cell membrane in both the directions. But more water will tend to move inside the cell compared to outside. Why more water will move inside the cell? Because the outside solution is dilute and this solution is known as hypotonic solution. What do we call this solution? Hypotonic solution. So in this type of solution, more amount of water will move into the cell compared to the water which is coming out of the cell. And what will happen to the cell size? It will automatically become big and the cell is likely to swell up. This cell will swell up and will acquire a bigger size. Clear? Now, moving to the second diagram and the second process. Here I am showing that I have taken this beaker in which I have added a medium of water which is exactly the same as is present inside the cell. Okay, means the medium is exactly the same. The concentration of water is exactly the same inside the cell as well as outside the cell. So, the movement of water will show that it is equal. The net movement of water across the cell membrane will remain the same. It is equal in both the directions. This condition or this solution is known as isotonic solution. Here, water do occur to move inside the cell. Water crosses the cell membrane in both directions, but the amount is going to remain the same. No excess amount of water is going to come out or move inside, and the cell will remain same inside. Okay, this solution is isotonic solution. Children, please pay attention. I'm talking about the solution and the condition of the cell simultaneously. Here the solution is hypotonic, that means dilute, and water, more amount of water tends to enter in. Here the amount of water, that means the concentration of water, remains the same outside the cell as well as inside the cell. So there is no net flow of water in either direction, and we say the cell will have the same size. It will remain as it is, and the solution is called isotonic solution. Clear? Moving to the third beaker. In this third beaker, again we are placing a cell as we have done in the previous two conditions. Here the medium has a lower concentration of water than the cell. It means it is not dilute, it is highly concentrated. That means low water concentration. When we say low water concentration, it means highly concentrated water. Right. It means a highly concentrated water cannot enter the cell, because here the water condition is dilute. Right. Same way, water has to move. Water will cross the cell membrane in both directions, but this time more water will leave the cell compared to compared to an excess which will enter the cell, right? More water will leave the cell than entering. Therefore, the cell will shrink. Here in this condition, the cell will shrink as it is losing water. This special case of diffusion is known as hypertonic solution, where this solution is hypertonic solution, in which the water, more amount of water will lose from the cell compared to its entry. Alright, so are the three conditions clear regarding hypotonic, isotonic and hypotonic, isotonic as well as hypertonic solution? Clear? Hypo means dilute, iso means same, water concentration same, and hyper means when there is low water concentration compared to inside the cell. So this topic is known as osmosis.